Thank you very much. It's great to be here. I'm going to run you through my background because the course that I teach is a little bit different from the research that I do in the lab, but hopefully I'll make it clear for you why there's actually a nice connection between my research and my teaching. So I did my undergraduate degree at Dalhousie University in Halifax, in Canada. And I focused on cell and developmental biology, but Dalhousie, has a really strong reputation in marine biology. And so I was doing cell and developmental biology in this environment where there were a lot of people going out and counting barnacles and collecting seaweed. And I was always really interested in that side of biology as well. After I finished my undergraduate degree, I went and did a master's in paleontology at the University of Bristol. Then I went on to do my PhD in organismal biology and anatomy at the University of Chicago. And I had sort of a really important and transformative experience for my research career. While I was a graduate student, I came to do the embryology course here at UNDL during the summer of 2007. And that was actually really important because, first of all, it turned me on to a research organism that would become the main research organism that I use in my research career, since then and still today. And also it built a sort of lasting connection with the MBL. And as David mentioned, I've been coming back every summer or autumn or both since then until I moved here. After I finished in Chicago, I did a postdoc in the Department of Physiology, Development, and Neuroscience at Cambridge, and then another postdoc in the Department of Biology back at Dalhousie. And then I actually started my research group at the University of Cambridge in the Department of Zoology. And although my lab was physically based in the UK, every summer I would pack up my lab and all my people, and we would fly over to Woods Hole, and my family would come as well, and we would basically spend our summers over here in Woods Hole at the NDL, doing experiments and working on marine organisms that are available here in Woods Hole. And there was one organism in particular that we were coming here for, and this was the little skate. So if you've been to the MRC yet on your visit, you will have seen many tanks of skates in the MRC, and those are our main research organs, and that's really what brought us here year after year. The skate is a relatively small cartilaginous fish, so a close relative of sharks and stingrays. And one of the things that makes the little skate a great model organism for cell and developmental biology is that they are, by cartilaginous fish standards, relatively small. So we can keep large numbers of them in tanks. But even more importantly, they're oviparous, so they're egg laying. And what you can see over here is one skate egg in someone's hand. You often find these cases washed up on the beach. They're called mermaid purses or devil purses. And inside that egg case is a single embryo and a ball of yolk. You can see the orange yolk through there. Now, if you take a look at how these embryos develop, we can open up the egg and carry out experiments on the embryos inside. Here you can see the embryo forming. It's a bit of a thickening of cells that will then elongate. This is going to be the head end up here. And this is going to be the tail end down here. And after about 4 or 5 weeks of development, we can actually take the embryos right out of the egg case and culture them in petri dishes or bowls of sand and seawater, and we can watch them grow. We can carry out all sorts of surgical manipulations, microinjections, these sorts of things, and then we can rear them through all the way to hatching. Although we don't actually need to rear them to hatching most of the time because they spend about 5.5 or 6 months inside this egg case. But actually by about 8 weeks of development, they formed all their major organ systems, their skeleton, their nervous system. And so we can carry out these kinds of experimental manipulations on the embryos and then look at the impacts on these different organ systems. And so after 8 years in the UK, I finally had the opportunity to move my lab here permanently, which is really exciting. And that's what we did last year, just in September. So I'm a relatively recent new faculty member, but I don't feel very new because we have been coming here since 2007. And so, I highlighted my sort of broad training because, although I've trained in cell and developmental biology, I always did so in the context of sort of, comparative evolutionary biology, trying to understand the origin of anatomical novelty. And one of the reasons why we're so interested in studying these shark and skate embryos is because in the field of developmental biology, there are a number of really well-established experimental model systems. So these are animals that are, really powerful because they have a range of tools, a range of resources, they're fast to develop, and these are things like the zebrafish, the frog, the chick, and the mouse. But the important thing to note is that all of these organisms fall within one group of vertebrates, which are called the bony fishes or bony vertebrates. And so if we're interested in understanding some of the earliest events in vertebrate evolution, so for example, the origin of things like jaws and paired fins, we need to have data from this other lineage of vertebrates, which is the cartilaginous fishes, and this is the sharks and skates. And only by comparing data between all of these different model systems can we really infer conditions that were present in the last common ancestor of these jaw vertebrates. And so that's why we've been focusing on studying the embryonic development of cartilaginous fishes and the embryonic development of the skate in particular. And so using this model system in my lab over the past several years, we've been addressing questions related to the development and evolution of jaws, the development and evolution of paired fins, the development of the vertebral skeleton and how the vertebral skeleton has evolved and how it's regionalized into distinct functional regions along the axis of the animal. And more recently, we've actually started focusing our research efforts on aspects of post-embryonic development, studying cartilage development and repair and growth in adult skates, and also the development of neuroendocrine cells, which are cells that release peptide hormones systemically in response to neural input and that are really important for regulating homeostasis and growth and repair of the skeleton. And so, you know, a lot of my work is in the area of cell and developmental biology, but like I said, it's always been sort of in this comparative and evolutionary context. And I'm also very passionate about biodiversity and nature. I spend a lot of my time taking pictures of nature, counting nature, wherever I am, whether it's in the UK or here on Cape. And so when Linda asked if I would be interested in teaching a biodiversity course for my teaching here at the MBL, I was really thrilled to say yes. And so that's what I'm doing now. I'm actually teaching this course right now, literally right now. We're at the very end of our first week. And I'm looking forward to hosting you all or whoever is coming on the tour this afternoon in my course to be able to come and see what we're working on. But this course, really like the other courses you've heard about and will hear about, this is a really hands-on, you know, based course. It's a 3-week course. In the first week, the students spend a lot of time in the lab learning key techniques for recognizing and counting and quantifying biodiversity. So this is a combination of more traditional morphological techniques, so using anatomical keys and imaging to identify animals that they collect from the field, and also using molecular methods like DNA barcoding to allow us to resolve species when it's perhaps difficult to do that using morphology. And a lot of the fieldwork that we do for this course is based around the salt marsh ecosystems that are, of course, really sort of a signature feature of the Cape. So this is Little Sibuisit and Wokoit Bay. Now, once the students have a strong foundation in these techniques for identifying and quantifying biodiversity, they design their mini projects with support from the faculty, and we sort of transition over the course of the module into more independent research, where the students are designing courses, projects, going out into the field to collect their data, analyzing their data in the lab, and all the way through we are peppering the course with lectures on key topics, so they do get the strong overview of, you know, the diversity of life and you know, important issues in biodiversity and conservation, but with a very strong focus on the marine biodiversity that we have here in the Woodsville area. And so that's basically what we do for the course. At the very end, the students are assessed based on a presentation of the results from their research project, which they make to their course mates and also to the broader MBL community. So anyone is interested in community presentation as well. And so with that, I'll end and hand over to the next speaker.